All right. Well, I guess we'll get started. Welcome to another DOE Isotope Program user group meeting. Previous group meetings have been on Actinium 225, Astatine 211, and Lead 212. Today we'll be having the Copper-67 user group meeting, and I will be the moderator, David Rotsch from Argonne National Laboratory. Now, the purpose of these meetings is to encourage free discussion and collaboration amongst users of these isotopes and accelerate their widespread use in medicine. So let's advance the slide. Here's the agenda for today. I'll be giving a quick 10-minute overview, and then we'll have a talk from Jack Shively from City of Hope, Alan Packard from Boston Children's Hospital, Brian Zeglis from Hunter College, Jennifer Bartels from University of Alabama, Birmingham, and Shaemus Gleason from Clarity Pharmaceuticals. And then we'll have a moderated Q&A session at the end. So we all know that medicine has been moving towards more of a personalized medicine through the combination of diagnostic and therapeutic agents. And the recent FDA approval of the Gallium-68 lutetium-177 Dota has really paved the way for other isotopes to make their way into the market. Now, another one that's exciting that we're aware of is the Actinium-225, of course, and that one is right now being paired with Gallium-68. However, there is a need and desire to find better suited pairs between the therapeutic and diagnostic agents. So Gallium-68 is a matched pair to actinium and lutetium, but their chemistry isn't identical, and the biodistribution isn't necessarily always the same. So, for example, with Actinium-225, a newcomer is the 132 cerium, where we can have a little bit better behavior or similar behavior. Then if we want to get a little bit more identical, we look at isotope pairs. For example, the PET imaging agents, scandium-43 and 44 with the diagnostic scandium-47. And of course, today, the Copper-67 pairs well with Copper-64. So Copper-67 is a theranostic agent. It has about a 2.5-day half-life, which makes it suitable for international shipments, but then also for the biodistribution for cancer treatments. It has a very nice average beta emission around 140 keV, and a very imageable gamma emission around 184 keV. So that's why it's a true theranostic isotope, where it can do both therapy and be used to image. And like I said previously, it pairs with Copper-64, and Copper-64 has been around for a while. So we've done a lot of work with, and we, the community, has done a lot of work with different chelates to find out how to sequester this thing appropriately and make certain that it doesn't go freely inside the body. For example, there's the Nota and then some cross-bridge cyclams. But more notably are the sarcophagenes that Clarity Pharmaceutical has been pushing through the clinical trials. And I'm very excited to hear their talk later today. Now, making Copper-67, we can make it with protons and neutrons and even with heavy ion fragmentation. Now, one of the problems that we've seen in the literature is that the specific activity hasn't really been very high. And depending on what that specific activity is, there's some challenges in radiolabeling. So why is Copper-67 so difficult to produce while Copper-64 is so readily available? Well, there's a few reasons for this. One, the production rate of Copper-64 is higher than that of Copper-67, but mostly the targetry is very challenging with producing Copper-67. We're working with zinc targets that have relatively low melting points, while the nickel target for Copper-64 has a relatively high boiling point and can withstand high beam currents. The other challenge here is that zinc usually wets in alloys with most metals. So think about everything that's been galvanized with zinc. So it makes it challenging to work with zinc. At Argonne, we've been looking at the photonuclear production of Copper-67, where we take high energy electrons, accelerate them down a beam pipe, and then strike them on a a converter, and then the electrons are transformed into bremsstrahlung photons that then interact with our target. And in these types of reactions, what you can get are you can knock off a neutron, proton, or even an alpha, or a combination thereof, to produce your radioactive isotope. The interesting thing here is that you end up with the bremsstrahlung continuum, where you have the photons with the energy of the incident electron, and then all the energy is all the way down to zero. So you get this cascade effect. So it's a little bit more challenging to get very precise production windows. Regardless, we've been producing copper-67 through the gamma-P reaction on zinc-68. You can also produce copper-67 from zinc-71 with the gamma-alpha reaction. However, that cross-section is relatively low in comparison to that of the gamma-P reaction. And what we've been finding with these types of reactions is that the theoretical database seems to underpredict what the actual cross-section is. Now, with this production route, it's important to use enriched targets, and zinc-68 is about 20% abundant, so it's a relatively expensive target material as well. So a lot of challenges with the zinc target material. With our production route, we produce roughly around 2 curies at end of bombardment, and this is shipped as a solid copper chloride to DOE Isotope Program and NITC customers. Copper-7 is identified through the 93 and 184 keV gamma emissions, and some of the purity specs that we have as minimum specifications are greater than 99% radionuclidic purity, greater than 50 curie per milligram specific activity. On average, our specific activity has been over 100, which has been really good. And we've had excellent radiolabeling results with Teta, Dota, and even Micosar. And this is part of the quality assurance and quality control that we have for our product, where we'll measure the apparent molar activity under these conditions with these isotopes. And anything over around 50 curie per milligram, typically we end up with greater than 1 millicurie per nanomole radiolabeling efficiencies at time of labeling, which typically occurs around 1 half-life after the end of bombardment. We've been able to send this out to collaborators for independent quality evaluations, where the University of Wisconsin was able to radiolabel NOTA and Dota with very good AMAs. And then the University of Alabama, Birmingham was able to radiolabel dotatate. The only consideration here was that they needed to increase the temperature. And once they got to 90 degrees Celsius, they essentially got 100% or quantitative radiolabeling. And Jen Bartels, I believe, will talk a little bit about this work later today. We were also able to perform some demonstrations of the imaging capabilities of copper-67 with the University of Iowa, where they took our copper-67 and injected it into a Society of Nuclear Medicine and Molecular Imaging Clinical Trials Network anthropomorphic chest phantom, where each one of these nodules was filled with copper-67 and then imaged with a human SPECT scanner. And then they used a low energy, high resolution collimator and a medium energy collimator to determine what was the best collimator to get the best images. This was then compared to PET image of copper-64, and then a SPECT image of Tec-99m, and then also a SPECT image of lutetium-177. The bottom line here is that PET obviously wins in the image quality. Tec comes in second, and then both copper and lutetium show the ability to image cancers. And with that, I would like to thank you for attending again. If you want to find out more information about copper-67 or other isotopes, please visit isotopes.gov. And I'd like to open this up to the other speakers and maybe start this in the Q&A section at the end. But one of the things that we found is that with our test batch that we sent out near the very beginning, had a specific activity around 50 curie per milligram. And all of these test batch recipients had very good radiolabeling. And so I'm curious as to what is the ideal specific activity that the community needs for these types of radioisotopes. Now, to give you an idea, if we look at the copper-64, copper-7, and lutetium-177, and we look at their specific activity based on the number of cold versus radioactive atoms, and then compare these, we see that 200 curie per milligram specific activity copper-64 is equivalent to approximately 40 curie per milligram copper-67, which is again is equivalent to approximately 18 curie per milligram lutetium-177. So I'd like the speakers to think about this while they're presenting, and maybe we can have a discussion towards the end. And with that, I'd like to introduce Jack Shively from the City of Hope, who will give our first presentation. And for all questions that come up, please use the QA function, write them in there, and then we will answer them in the QA session at the end of all the presentations. Thank you. Jack, do you want to share your screen and start presenting?
So I'd like to thank the organizers for giving us an opportunity to present our experience with copper-67. My involvement began with a phone call from Steve Sugden. Probably a lot of you, the same thing happened. And of course, the idea was to start getting regular shipments from Argonne, as Dave just explained. And we start out by just doing test labels on peptides and antibodies that were dotylated. And these were done through our radiopharmacy, which is run by Kofi Poku. And then finally, I'll talk about some preliminary animal studies that were performed by a graduate student and now postdoc, Megan Nix. Okay, so just for maybe historical purposes, I know a lot of you got copper-67 in the past from Idaho State. This was one of our shipments that we got back in 2014. And we were able to get a fair number of millicuries from them at the time, and it gave very good labeling. The main problem was sporadic shipment and problems with labeling. So the first shipment that we got from Argonne National Labs came in 2018, but we didn't have very good luck with that for either the peptide or the antibody labeling. And then you can see that throughout 2019 and 2020, we got a number of shipments, and both the peptides and the antibodies all labeled basically quantitatively. And this shows the labeling ratio that we were using for peptides. It was always 10 millicuries per milligram, and for antibodies, it was 10 millicuries per milligram, and for peptides, 10 times that 100 millicuries per milligram. So our usual targets were in the range of like, for peptides, 200 microcuries for 2 micrograms for imaging or therapy. And for the antibody, that would be a target of 1 millicurie labeling 100 micrograms. And so based on all these successful test labels, in August of 2020, we decided to go ahead and do an animal study using the peptide. And again, it labeled essentially quantitatively. Let's see, I think, Dave, you didn't show many of these, but each shipment came with a certificate of analysis, and you can see that they tested it versus TITA, Dota, and AMSAR, all labeling to high specific activities, along with all the impurities. So that's one of the things that you get with the shipments. And of course, these were tested first by ITLC labeling on dotylated antibodies. So this is one of the antibodies that has gone into the clinic here at City of Hope quite a bit. It's an anti-CEA antibody. It's humanized. We call it M5A. It's dotylated, and these are all clinical grade. And so what we did is we labeled the copper-67, and this just shows you that the ITLC basically is essentially quantitative labeling. And then we also take the sample and we run it on size exclusion HPLC to see how it lines up with antibody, which it does. Then you can see a small peak that's due to the chase with 10 millimolar DTPA. And so these also carried out for stability. And so the stability carried out to say 5 days basically shows no degradation, no loss of copper-67. And down here in this corner, you can see an example of an ITLC for a peptide, a Dota-related peptide, which I'll talk more about. And again, you can see it's quantitatively labeled. This is also confirmed by reverse phase HPLC to show that, that the peptide has picked up the copper-67 and is stable. And again, here are the conditions for labeling. So these are our standard conditions for copper-64, and we just carried them over for copper-67. And so our small peptide target that is actually now GMP produced and is in the vial, and we're waiting for working on an IND to get it into patients. I think our first target will be neuroendocrine cancers. But in this particular example, they were put into tumor-bearing animals with a prostate tumor. This is called PC3. And PC3 does not express the neurotensin receptor, so we transfected it. And basically now it expresses 100% of the neurotensin receptor, which can bind neurotensin. This just shows you over here that compared to bona fide neurotensin 8 to 13, essentially only a slight loss in activity, binding activity for the peptides. And the synthesis was performed by solid phase, included a number of unusual amino acids to stabilize the peptide in serum. And we added a lysine at the N terminus. And during solid phase synthesis, we coupled in Dota, so it's on both the alpha amino group and the epsilon amino group. And we did this in a way to hopefully increase our chances for higher specific activity, which is needed for getting a high specific activity peptide product for good labeling and good imaging. This is all published in Cancer Biotherapeutics Biopharm last year. And so down at the bottom, you see a copper-64 image in the nude mice at 2 hours and 4 hours. And you can see that, that the tumor has basically a very strong background. Blood was less than 1. And the kidneys are, as you might expect with the peptide, are the biggest confounder in these types of studies. And so that's something that also was true for the somatostatin analogs that most of you are familiar with. We also were able to perform time activity curves, and this just shows you that rapid clearance from the blood and basically uptake into the tumor is complete within, let's say, 10 to 20 minutes and is more or less stable. And the ratio of tumor to blood is about 5 to 1 in the nude mice. We got a surprise. We did some studies in NSG animals, and the kidney uptake was higher in these animals. So this is an interesting point. It may be that there's some variation in neurotensin uptake depending on the animal model. At least with the nudes, the uptake in kidneys is very rapid within a couple of minutes, and then it decays very rapidly down to say maybe a reasonable background by 1 hour. So as a result, on the last shipment that we got from Argonne National Labs, we decided to do a therapy study, based on our success with both copper-64 and gallium-68 imaging in these mice. And so we let the tumors grow for about 10 days and then began RIT. So this is the copper-67. And we wanted a high dose in this particular study because of the rapid clearance of the peptide. So we did 125 out to 500 microcuries. And this is the result. Unfortunately, this is a disappointing result. You can see that we don't really have any dose response and any real response to therapy in this particular study. So this kind of threw a damper on us continuing with copper-67 in this particular model. That certainly doesn't mean that it can't perform in other peptide models, and we'll probably hear more later on. And it may have something to do with the specific activities. We may have to go to higher specific activities to get some therapy. The good news was that there was no weight loss over this range all the way up to 500 microcuries in the mice, and no other indications of toxicity. So I can say it was safe, it just wasn't successful in terms of tumor reduction in this model. These are the individual mice for this particular study. There's, basically they all look the same. And then I'll just end with, before we got the Copper-67, we had done an actinium therapy study in the same model. And there we did have a dose response. So I think that the model is accessible to targeted alpha therapy. And we're still, the jury's out for whether or not targeted beta therapy versus alpha therapy can compete in this model. But at the moment, we're very interested in going down the actinium road. But I know that's probably not the right format for this. So that's the end of my slides.
All right, thank you very much, Jeff.
Sorry about that, Dave.
Oh, that's all right.
I thought some negative data might also spur the audience along too.
We definitely appreciate that. All right, next we have, and again, we're going to reserve questions until the end. But next is Alan Packard. Alan, if you want to share your screen.
There we go. That's better. Okay. Thanks very much, Dave, for putting this together. This is great. I've been working with copper since I was a grad student in Copper-64 since the 1990s, and I've been trying to get my hands on Copper-67 since shortly after that. So these recent advances in the production of copper are very exciting to me. I'd like to start by talking about, let's see, next slide. It's not going next slide. We'll try plan B. Okay, start with disclosures. This is our work was funded actually by Clarity Pharmaceuticals. So I guess Clarity has something of a presence here today. We're interested in neuroblastoma. It's the second most common solid malignancy in children. There are a relatively small number of patients, which is a good thing. We don't want a lot of kids with cancer. About 650 to 700 new cases a year in the United States. Unfortunately, what this means is, from the clinical trial point of view, is that it's relatively difficult to enroll these patients in clinical trials because no one hospital gets a significant number of these cases. The average age at first presentation is about 19 months, and the disease is frequently diagnosed at later stages, stages 3 and 4. And the reason for this is a lot of the symptoms, the early symptoms of neuroblastoma mimic a lot of other diseases of childhood. So you don't go looking for neuroblastoma, you look for things like flus and stomach bugs. Early treatments are effective, but the patients frequently relapse, more than 75% relapse with widespread metastatic disease. And the survival rates of patients with relapse disease is extremely low. And the question we're looking to address is it possible to improve the outcome of these patients? There are several possible targets in terms of treating neuroblastoma. We've been looking previously at GD2, it's disialloganglioside, that's expressed on essentially all neuroblastomas. We're looking at that with the 1418 antibody family. You can also use MIBG, which targets the norepinephrine transporter. More recently, efforts have shifted to SSTR2, the somatostatin type 2 receptor. That's present on up to about 90% of neuroblastoma tumors. It's targeted with an octreotide derivative. This has very high binding affinity, and it's a peptide, so it's relatively easy to modify in terms of putting a chelator on it. Of course, Gallium-68 dotatate, that spot is already approved for imaging adult somatostatin receptor positive neuroendocrine tumors, but it's not been extensively evaluated in neuroblastoma. And lutetium-177 dotatate, Lutathera, has been approved for treating adult somatostatin receptor positive neuroendocrine tumors. And again, it hasn't been extensively evaluated in neuroblastoma. So if we have dotatate, Gallium-68 dotatate, and lutetium-177 dotatate, why do we need another agent? Well, starting with Gallium-68 dotatate, first is the high-cost Gallium-68 generator. If you're a pediatric hospital like we are at Children's in Boston, we don't have a lot of patients coming through, so we can't really afford to have a generator in-house for the occasional patient who comes through with neuroblastoma. Another issue is the availability of the generator. Germanium-68 is in somewhat short supply, so the generators are hard to get. The lower resolution of the Gallium-68 images, I'll talk about that a bit more later. And the short half-life of Gallium-68, 68 minutes, limits the ability to do dosimetry calculations, which is becoming increasingly important. You can always do dosimetry calculations after the fact, after you've done the first dose using the imageable photons from lutetium, but that doesn't allow you to predict what your first dose should be. And in terms of lutetium-177 dotatate as a matched pair or semi-matched pair to gallium-68 dotatate, it isn't really a matched pair. And as Dave mentioned in his introductory remarks, there are some differences in the biodistribution between gallium-68 dotatate and lutetium-177 dotatate. So looking a bit more closely at gallium-68 and copper-64, as I said, gallium-68 has a 68-minute half-life versus 12.7 hours for copper-64. That means you really can't do dosimetry estimations with gallium-68. It's very easy to do that with copper-64. The beta plus yield for gallium-68 is great, about 90% versus 18% for copper-64. That advantage disappears pretty quickly, though, because of the short half-life of gallium-68. If you do the calculation about how much is left as a function of time, the crossing point is about 2.5 to 3 hours. So if you're imaging after that time, copper-64 actually gives you more photons. I mentioned before the high positron energy of gallium-68, 800 keV versus 278 keV for copper. That's about the same as F18. So the image resolution is about the same as you see with F18 images. And that high energy of the positron means that the positron range is about 4 millimeters versus 1 millimeter. I mentioned before the production from the generator. More recently, because of the shortage of the generators and the cost, people have been making gallium-68 in-house on a cyclotron. But that, of course, means you have to have a cyclotron so you can make it. Production of copper-64 is relatively simple, cyclotron produced. And because it has a longer half-life, it's relatively easy to ship versus gallium-68. You can ship it short distances, like within the city, but you can't ship it across country like you can copper-64. The disadvantage of copper-64 is that it's very labile. Somebody alluded to that earlier. I don't know if it was Dave, but the problem is you have to have a chelator that it sticks to, and that's been a historical problem with copper-64. In terms of the positron energy, whether it makes a difference, this is some work done by Andreas Jaeger's group in Copenhagen. They are the first people to make copper-64 dotatate. They basically just stuck copper-64 in a dotatate kit to see what would happen. The top image is gallium-68 dotatalk, and the bottom image is copper-64 dotatate. And if you look at the image here in the liver, you can see these two spots here and the larger one here in the liver. And in the lower abdomen, you can see a lesion here and a larger lesion here. But if you look at the copper images, these two lesions here are much more clearly defined, and you can see the structure of this lesion a little bit better with the copper-64. And if you go in the lower abdomen, you can clearly see these three small lesions here that are sort of barely visible in the gallium image. And this one is better resolved in terms of it having a lobe over here that isn't really visible in the gallium-68 image. So clearly, in these images, we required an hour post-injection. So it's not just waiting for the gallium-68 to decay. It's the fact that the image resolution is superior. In terms of the chelators, as Jack and Dave mentioned, a lot of work was done with Dota. The Dota core, this is the crystal structure of the copper Dota complex without the PBA tail that's been added subsequently for binding to proteins. It's easy to make the complex, just put in an acetate buffer at room temperature. Problem, of course, is the copper is lost from the complex in vivo very quickly. And if you look at the structure, it's sort of obvious why this happens. The copper is sitting, this is the sort of backbone of the 4 nitrogen cores. The copper is sitting above that. It's got a couple of carboxylates, but these carboxylates are very labile, so they're flopping on and off. I'm waving my hands here, showing you the carboxylates flopping on and off. I keep forgetting, nobody can see me. And it makes that copper very accessible. This is the methyl CoSAR. This is the ligand that Clarity is using. This is a derivative of diamsar. Diamsar is just a bare amine. There's an amine here and amine here. Paul Donnelly made a series of derivatives of this, Paul Donnelly at Melbourne, and they came up with this derivative to use for their clinical trials. In this case, the nitrogen on this side has been functionalized with an ambit and a carboxylate out here to bind to the proteins. As with Dota, it's relatively easy to make it. You just put in an acetate buffer at room temperature, and the copper just slides right into the core. You can see this core is pretty cool. There's a nitrogen here at the top. You look at chelate rings, here's one of them here. There's one here coming towards you in the front, and then there's another one going in the back. So that copper is really buried in the middle of that. If you look at the space filling model, the copper is really stuck in the middle of that. So it forms very stable copper complexes. So if we're looking at therapeutic radionuclides, that things get a little more iffy here. What's the optimal beta energy? There are obviously a range of beta energies out there, lutetium-177 and copper-67 at one end, and things like yttrium-90 with a 1 MeV beta at the other end. One thing you do want is no extraneous emissions. You don't want like a 1 MeV gamma ray out there that's radiating people without contributing anything to either an imageable gamma or to the radiation dose to the patient. You would like an imageable gamma. Early thought was that you probably didn't because you didn't want this background radiation getting out of the patient, but more recently as interest in dosimetry has increased. People are more interested in having an imageable gamma, especially with SPECS CT cameras becoming available, which makes it easy to do dose calculations for single photon emitters. And what's the optimal half-life? That's an interesting question. You want it somewhere compatible with the half-life of the vector. Being a peptide, you want it maybe a little shorter, and antibody maybe a little longer. And the ultimate cost question with all of these is cost of availability. If you can get a great radionuclide, but it's so expensive you can't afford to use it, that doesn't do anybody any good. And availability is obviously an issue, as both Jack and Dave alluded to. Copper-67 was very difficult to get until recently. So it's very much stymied the ability to do any studies of therapeutics with copper-67. So comparing lutetium-177 and copper-67, the half-life for lutetium-177 is 6.6 days versus 2.6 days for copper-67. Beta yield for both of them is 100%. The beta energy is about the same, which means their beta range is about the same. They both have imageable gammas, lutetium-177 perhaps less so, but as we saw in the images before, the 185 keV gammas from copper-67 yield very good images. And as I mentioned before, the question is production. When lutetium-177 was first being used, it was made from an N-gamma reaction on lutetium-176, which gives relatively high specific activity material for an N-gamma reaction. That's because the cross-section is very high, about 300 barns. So 1.1 terabecquerels per milligram isn't too bad, but more recently, people have been making it from a terbium-176 to terbium-177, and that decays to lutetium-177. So you basically have a generator and a terbium-lutetium generator. And the activity, the specific activity of that material is about 3 terabecquerels per milligram. Dave talked a bit about the yield of the specific activity of copper-67. I won't go into that in too much detail other than to say that the specific activity of the gamma P stuff is about 15 terabecquerels, much better than the 1.9 terabecquerels we were getting before, although either of these is fine for labeling peptides. So in terms of answering the question about neuroblastoma, the first question we have is, does copper-64, copper-67 sartate accumulate in neuroblastoma liver metastases? And is copper-67 sartate an effective treatment for metastatic neuroblastoma? And the question that Jason and I have been thinking about a lot is, if we can treat the disease early enough, can we actually prevent the development of metastases? If you wait until the metastases develop, it's much harder to treat them, and the outcome is much worse. And we've dubbed this prophylactic radionuclide therapy. We have some other names for it, but this is probably the most the less troublesome, shall we say. In terms of creating a metastatic model of neuroblastoma, we do this in nude mice, do a dorsal incision, excise the spleen, or expose the spleen, inject the spleen with 1 million IMR32 neuroblastoma tumor cells, wait 2 minutes post-injection, then perform a splenectomy. We have to do this because if we don't, the disease will develop in the spleen and kill the animal before the mets have a chance to develop. Close the incision, and about 2 weeks later, we have 1 to 2 millimeter mets present in the liver. Here's an imaging study to validate that we actually have this. This was done with copper-64 sartate to validate that the tumors have become established. This was carried out through 3 weeks post-inoculation. And you can see at 1 hour post-injection, you can see a little bit of activity here starting to accumulate in the liver. Most of the activity is in the bladder. You can see it in the transverse sections here. At 5 hours, you can see a very clearly defined spot here, and again in the transverse sections. 24 hours, that spot's still there. You can see some other spots beginning to develop here. A lot of activity in the gut as it's being excreted. Look at the transverse section, you can see a couple spots here. And at 48 hours, you can see this spot still and a couple other smaller spots. And in the transverse section, you can see a lot of spots here. We sacrificed these animals and excised the livers and did H&E staining and autoradiography to confirm that, in fact, these lesions were neuroblastoma. The other thing to note is, in contrast to what you might see, especially at 24 and 48 hours, if you use Dota as a chelator, there's basically no copper activity in the liver except in tumors. So the therapy protocol, we treat the animals with copper-67 sartate. This was a single treatment. It's done in the first experiment, 4 weeks post-inoculation. We treated the animals with 2 different doses of copper-67 sartate, 250 microcuries and 500 microcuries. And then we did a second experiment 2 weeks post-inoculation where the tumors were smaller. This was a single dose of 500 microcuries. And here are the Kaplan-Meier graphs for this. On the left is the 4-week tumor growth study. Control animals here. Some of them outlived the treated animals. The low dose animals died fairly quickly, and the high dose animals died a little less quickly. But if you look at all of this in terms of statistics, there's no statistical difference between these 3 treatments. If you back it up to 2 weeks, though, when the when the tumors are smaller, there is a significant difference between the control animals and the high dose animals. This is 500 microcuries. And you see about a 30% increase in survival for the treated animals at the higher dose. So in fact, if we look at small tumors, we can effectively treat these, increase survival by about 30%. So summarizing this, in fact, we can image small 1 millimeter liver mets with copper-64 sartate. Copper-67 sartate extends life in mice with smaller tumors, those that have only been growing for 2 weeks. Copper-67 sartate is more effective in treating smaller tumors than it is in treating the larger tumors that are present at 4 weeks. So this still leaves us with a bunch of questions. What about lutetium-177 dotatate versus copper-67 sartate? Is lutetium better or worse than copper-67? Are higher doses of copper-67 sartate perhaps more effective in these smaller tumors? We only went up to 500 microcuries. And the issue that Jack raised, are alpha emitters perhaps more effective in treating these small lesions than beta emitters? We don't want to raise that question when we're talking about copper-67, but somebody always asks when we send papers in. And perhaps are antibodies better vectors than peptides? We know that when you treat animals with antibodies, the uptake in the lesions is very high compared to what you see in peptides. So maybe an antibody might give us a better therapeutic effect. And again, going back to the original question, if this treatment is effective, if we start even earlier, can we actually prevent these tumors from growing? So what we'd have to do is treat the animals shortly after we injected the IMR32 cells and see if we could actually prevent them from taking in the liver mets. So this goes back to the concept of prophylactic radionuclide therapy. So as I said several times, a lot of this work, most of this work was done by Jason Dilling, who's here today. And it was supported by Clarity Pharmaceuticals and the Children's Hospital Radiology Foundation. And thanks again for including us in the symposium today.
You know, Alan, that was a very interesting talk. I will have some questions for you.
Uh-oh, ask Jason.
Next, we have Brian Zeglis from Hunter College. Whenever you're ready.
Hi, everybody. I'm Brian Zeglis. I am an associate professor of chemistry at Hunter College, and I've got appointments in the departments of radiology at Memorial Sloan Kettering and Weill Cornell. And I think I'm going to skip the kind of introduction to copper-67 because Dave and Alan and Jack have done such a great job on that. And I'm going to kind of jump into exactly what we've been trying to use the copper-67 for. And in our case, that's using it in conjunction with copper-64 to create a theranostic approach to in vivo pre-targeting. So in vivo pre-targeting is kind of founded on this idea of antibodies as vectors for radionuclides to tumor tissue. And so antibodies are great in a lot of ways, right? So they are specific, they have high affinity, they have high stability, but they've also got their problems. I mean, we know they can be immunogenic if they're not humanized or fully human. They can only target extracellular targets, and unless you do some of the cool things that Bart Cornelison is doing with nuclear targeting peptides. Their construction can be precise without site-specific modification. And finally, and most importantly here, is their slow pharmacokinetic profile. So antibodies can take anywhere from a day to 3 days to 5 days to 10 days to get to their optimal biodistribution in the body. And if we're trying to do radiotherapy with that, or PET imaging, there's this huge chunk of time where the patient is getting an unnecessary radiation dose from the antibody or the radioimmunoconjugate before it's reached the tumor site. So what we've wondered, and others have wondered in the past, is can we create a system that has all of the targeting advantages of full-length antibodies, but the pharmacokinetics of small molecules. And so one way to try to harness that you've probably seen are using antibody fragments, but antibody fragments don't have their own set of problems. So we've turned to this idea of pre-targeting. So pre-targeting is this idea that we can decouple the antibody and the radionuclide, inject them into the patient separately in 2 separate injections and allow them to recombine. And so how this would work is we start with our tumor, we do an injection of an antibody, but this isn't any antibody. It's a special antibody that's modified with this little yellow ball here, and that yellow ball is going to be important later. So that, that antibody accumulates in the tumor and clears from the blood. And then after a couple days or 3 days or 5 days, and when it's accumulated at the tumor, we inject the radioligand. And the radioligand is now a small molecule. And it has this other special ball here. And these, in the yellow ball and the pink ball, can come together in vivo and do some sort of ligation. And so this small molecule radioligand flies through the body relatively quickly, binds to the antibody that's already on the tumor, thereby doing radiochemistry at the tumor site, or clearing from the body quickly. And so you end up with a properly radiolabeled antibody, tumor site. And so the big question for years was what are these 2 species that allow for the in vivo ligation? People have tried streptavidin and biotin, people have tried bispecific antibodies and radiolabeled haptens, and people have tried oligonucleotides. And I'd make the argument, and I would if I had a longer talk today, that the first is too immunogenic, the second is too complex, and the third is too unstable. So we came along about a little less than a decade ago, and we're excited about this reaction, the inverse electron demand Diels-Alder reaction between this guy here, a tetrazine, and this guy here, a transcyclooctene. And these come together in extremely quickly, second order rate constants of 30,000, 100,000, 150,000 inverse molar, inverse second. They're modular, meaning we can attach either of these 2 things to anything we want. And most importantly, they're bioorthogonal, meaning in the sea of functionality that is the biological milieu, they'll only react with each other. So how would pre-targeting with this reaction work? Well, we'd start with an antibody labeled with a transcyclooctene. We inject that into a tumor-bearing mouse or a human with a tumor. It takes time to accumulate at the tumor and clear from the blood. Then we inject a tetrazine-bearing radioligand. It travels through the body quickly now because it's a small molecule, clicks with the antibody at the tumor site, or rapidly clears. So we've done a lot of work over the last 7, 8, 9 years, developing both pre-targeted PET imaging systems using isotopes like F18, Gallium-68, and Copper-64. And we've also done a good deal of work optimizing pre-targeted radioimmunotherapy approaches, like using lutetium-177 and Actinium-225. But the holy grail here was be able to create a theranostic system in which a single species for the radioligand could be labeled with 2 different isotopologues and could be used in series. And that, of course, is the idea why we turned to Copper-67, because we could use Copper-64 for a PET imaging component of a theranostic system, and then Copper-67 for the therapeutic part of the theranostic system. And so we built this system a couple of years ago. This was all pioneered by my postdoc, Dr. Outi Kainanen. The antibody that we've based this all on is the HUA33 antibody. It was developed by the Ludwig Institute, that is now the Olivia Newton-John Cancer Research Institute. The antigen is the GPA33 antigen. It's expressed by the vast, overwhelming majority of colorectal carcinomas. And the important thing for us is the antibody, when it's bound to its antigen, is not internalized rapidly. It stays on the surface of the cells. So we built our system. We took an A33 antibody and we tacked on 3 or 4 or 5 transcyclooctenes. And then we built a radioligand. And the radioligand here is the tetrazine with a short linker linked to the MECASAR chelator that we could label either with Copper-64 or Copper-67. Of course, the latter is for PET imaging, the former is for PET imaging and the latter is for therapy. And so this is how we envisioned this theranostic system working. So we start with our animal with the tumor, or our human with the tumor. We inject the antibody labeled with the transcyclooctane, and it accumulates at the tumor and clears from the blood. And then we inject the diagnostic radioligand. I can either click with the antibody at the tumor or clear. And then at this stage here, we can do PET imaging. And this is where this is where a decision tree is. If there's enough radioactivity at the tumor, showing that there's a lot of antibody transcyclooctane in the tumor, we could go on and we can inject a therapeutic radioligand, get an in vivo click ligation, and then do therapy that way. But if there's not, we could just say this isn't the appropriate therapy in this patient. So we ended up doing this. We did a pretty extensive longitudinal therapy studies. And so here are all the different cohorts. We had one cohort with just saline, one cohort with just the antibody, one cohort with just the copper-67 labeled radioligand. Then we did 3 different cohorts where we did the antibody with the transcyclooctane, waited 1 to 3 days, and then gave the copper-67 tetrazine. And we did 3 different doses. Then we got a little crazy with a couple more. We tried a fractionated dose where we gave half the dose after 3 days and then another half of the dose after 5 days. We also did a theranostic cohort where we inject a copper-64 tetrazine at 3 days and a copper-67 tetrazine at 4 days. And then we compared this all with the directly labeled copper-67 antibody that started labeled, so wasn't pre-targeted. And what we saw when we did this experiment in the mice was really heartening. So all 3 control situations did not help the mice at all. All of these are individual mice showing tumor growth over time. And in the saline, antibody only, and copper-67 only case, the tumors just grew and grew until we had to euthanize the mice. Then in the 3 different doses of the pre-targeting, we saw a dose-dependent effect. We see in the lowest dose of pre-targeting, well, the median overall survival is extended, but the mice all have to be euthanized. In the medium dose, 37 megabecquerels, it works a little bit better, but not perfect. And then in the highest dose, except for 1 animal, we got a curative effect over 200 days. And we can compare these to the fractionated dose, the theranostic dose, where we got the copper-64 and copper-67, and finally the radioimmunotherapy. And so if we look at this over a Kaplan-Meier plot, it shows a similarly pretty rosy situation for us, at least so far, is that we have, with the 3 different controls, saline antibody only and copper-67 radioligand only, our median half-lives are 21, 21, and 32 overall, median overall survival. When we go to 18.5 megabecquerels of pre-targeting, we get an extension to 69 days median over our medium dose, 99 days. And when we go to our highest dose of pre-targeting, we get over 200 days. And the same is true for the directly radio-labeled antibody and for the theranostic. And then we get 189 days for the fractionated dose. So this was all really heartening, but what really made us excited is the next slide. And so in the next slide, we didn't just inject the copper-64 labeled tetrazine like we showed you before, but we did an actual theranostic experiment. So in this experiment, we injected the A33 TCO. We waited 3 days and injected the copper-64 Micosar tetrazine. And then we did some PET imaging, and we used those PET images to quantitate the dose in the tumor. And then in all these animals, we then a day later injected the copper-67 Micosar tetrazine. And what we saw was really exciting. So here's an example of the PET images we see. We see overwhelming uptake in the tumor tissue and very little uptake after a few hours in the other tissues. And what was really exciting is when we plotted the absolute uptake of radioactivity in the tumor, which ranged from 3 kilobecquerels to 13 kilobecquerels about, we saw that the animals with the lowest dose that we observed in the tumor via PET were also the animals that did not respond to the therapy. So we've been able, with 2 isotopologues and a single pre-targeting system, what we've been able to do is create at least what in this small trial was a very effective theranostic, and that in each of these, with a threshold of about 6 kilobecquerels, we could have gone through this procedure and said, okay, well, This animal has 12 kilobecquerels of copper-64. Go ahead with the copper-67, they're likely to respond. This animal only has 4 kilobecquerels from the copper-64 PET image. It's not going to be worth it, and it's not going to be worth it to do the therapy. So we were able to, in essence, we were able to predict the response to therapy because of the PET imaging. So with that, I don't want to go over my time. We've also done some work with this in a canine system in which we used a bone targeting TCO and a copper-64 labeled sarcophageine ligand. This also worked to delineate osteosarcoma, and we're going to try to go ahead and do some copper-67 therapy in canines with osteosarcoma in the future. And so that's about all. So I hope I've shown you in this short presentation that in vivo pre-targeting based on bioorthogonal click chemistry is a promising strategy for getting all of the benefits of antibody-based radiopharmaceuticals without their pharmacokinetic drawbacks. We've demonstrated this system a lot. In this case, we're focused on the demonstration with copper-64 and copper-67 specifically, and the implications for radioimmunotherapy, particularly in concert with theranostic imaging, are the most exciting, I think. And so what's next? Later this year, we're going to start our clinical trial in pancreatic cancer patients with a pancreatic cancer targeting transcyclooctane immunoconjugate and a copper-64 labeled sarcophagene. We're particularly excited by the copper-67 results because then we could translate without new pharm tox studies and use a copper-67 labeled tetrazine SAR for future therapeutic studies. And that's it. So here are all the references for those of you who might be interested. I think these slides are going to be available in the future. And for acknowledgements, all the folks at Hunter, Sloan Kettering, Paul Donnelly at the University of Melbourne, Jeff Bryan and Heather Henkins at Missouri, who have been in charge of the canine imaging, and for funding, Clarity Pharmaceuticals, and a bunch of different NIH grants. And that's all from here.
Well, those are really exciting and promising results, Brian.
Yeah, it worked pretty well. We're pretty psyched.
I'm looking forward to reading all those papers now. All right, so our next speaker is Jennifer Bartels from the University of Alabama, Birmingham. Jennifer, if you'd like to share your screen.
Thank you for the introduction. And if you don't know who I am, I'm actually a staff scientist for Dr. Suzanne Lappy. She is away at ACS this week and has asked me to present on her behalf. So here I'm going to kind of change gears a little bit and give you a little bit of background about our facility, who we are, what we can do, and then we'll go into the little bit of copper-67 work that we did last year. So first, we're a relatively new facility. You're kind of young. We are part of the UAB Advanced Imaging Facility. We're located in the basement of the O'Neill Cancer Center. Our cyclotron is actually right under the sidewalk. So our facility, right outside our facility, you can walk down and there are 2 PET CTs, and then there's a PET MR, and then we have a metabolite area, but then our facility is right here. It's 20 feet away. So we have our quality control area. We have a standalone radiopharmacy where we have 2 nuclear pharmacists who draw up all of our IND drugs, as well as a few approved drugs. We have a radio production suite, which has stacks of mini cells, larger hot cells, and then it has around 6 or 7 automated synthesizers. But then probably where most of the magic happens here is in our research space, which is joint between our production staff, which I'm part of that, as well as our students. We have roughly 8 or so graduate students right now. And although we can't make copper-67 here, we do bring up a lot of fun and interesting isotopes that you can see here on the right. And of course, the bread and butter of our facility is our cyclotron, which we affectionately call Blue. It's a TR24 from ACSI. We have variable energy, upwards of 24 MeV, so a little bit more powerful than most facilities that you're used to possibly. And we can throw upwards of 300 micrograms on target, but we definitely do not go that high. We have the ability, we have dual extractions. There are 4 beam lines, and we have a variety of different targets that we can bombard on. on top of the traditional isotopes that you're all very well familiar with, we do make a lot of the other radiometals that have been presented in previous talks already. So this scandium 43, 44, 47, as well as titanium 45. We're just now getting into vanadium 48, manganese, cobalt, copper 64, and zirconium 89. So we are, our radiopharmacists have the ability to ship some of our IND drugs in a lot of the surrounding states. And then we also are DOT certified to ship radio metals around the country. We do mainly zirconium 89 and copper 64 right now. And we just now started sending some isotopes into Canada, but we all know FedEx is FedEx, and sometimes it gets there, sometimes it doesn't. We do have a lot of collaborative efforts right now doing copper 64, some in clinical trials, as well as some soon to be clinical trials here. And Susie also asked me to mention that we just became a member of the DOE University Isotope Network, and more details will be shared about that soon. So now what we're all here to talk about really is the Copper-67. So we received one package from Dave last year, and we tried to do as much with it as possible. So I'm really just going to show just a small snippet of what we did. For me personally, I'm a chemist, so my main goal was just labeling copper dotatate. So Dave showed this a little bit earlier, and then I'll show the animal studies on the next few slides. So as he mentioned, we basically had to increase the amount, increase the temperature to really get good labeling. But once we increased to about 90, we could get near quantitative yield. And using your normal silica gel impregnated TLC paper, it readily separates between copper-63, copper-67, and the labeled compound. So we did a quick proof of concept animal study. We have a strong collaboration with our small animal imaging core here. It's led by Dr. Anna Ceresi. So they take care of the small animal PET CT, which is located in the 6th floor of our building. But then we also have a small animal SPECT CT, which is in another building. And so basically the goal of this study, which came together in about a week, was just to do a quick study between the clearance of just free copper-67, see where it goes, and to also study the copper-67 labeled dotatate in a tumor-bearing mouse model. So Dr. Ceresi does a lot of studies with breast cancer imaging, and she happened, when we told her that we're getting copper-67, we want to label dotatate, she happened to have some animals that had 41 breast tumors. This is a metastatic, or compared to a metastatic breast cancer model that happens to express SSTR2. So we said, this is great. So we just did 2 mice. It's really short compared to a lot of data presented previously, but first we just wanted to look at a clearance of this free copper-67. This was QS in PBS. We did roughly 500 microcuries injected, and then we observed the clearance over time at day 0, which was probably about 30 minutes post-injection, and then 1 day, 24 hours and 4 days post-injection. And then for the tumor-bearing mouse model, we also did the same, roughly 500 microcuries of the compound of interest, and then did the same imaging time points, but then also added day 5 and day 6, just out of curiosity to see, because we weren't sure how long the compound would actually stay in vivo. So this is just showing the clearance of the free copper-67 in the naive mouse. You can see it really loves the liver. It's basically a sink. And then over time, it slowly goes from the liver down through the gut, readily observed in the coronal slices. Basically at day 4, there's not a lot of activity present, which is to be expected. As for the copper-67, I believe Dave kind of showed this picture very early on, which you may not have noticed, but I noticed because it's here. So we did dynamic imaging. Oh, forgot to mention, all of these slides are provided by Dr. Sharon Samuel, who is part of the staff in the UAB Small Animal Imaging Core. So dynamic imaging doesn't really show much. You really see more in the tomographic and as well as the chronoslice. So if you notice the circle that's highlighted down here, basically within 30 minutes of injection of the copper-67 dotatate, we actually see localization in the flank tumor. This is a sub-Q flank tumor. And as a comparison, just to show you, it actually does have a slightly different clearance. We see mainly kidney uptake and bladder excretion, and then over time you start to see more liver uptake. And but it is just a proof of concept. This is just one mouse study, but the good thing is like we proved our point. We can see uptake with this compound in this mouse model. And so we're really hoping in the near future that we'll be able to explore more copper-67 therapy studies in perhaps in this mouse model again, or in other various preclinical models and different compounds. So just short and sweet, I have to thank Dr. Laffey. This is Dr. Laffey, as well as Dr. Ceresi. And these are our groups. We're a pretty big group. We do a lot of science. And then this is a small animal imaging port. And of course, Dave for sending us some copper-67. And that's it.
All right. Thank you very much, Jennifer. Yeah, I snuck some of those images into my.
It's okay.
All right. Next, we have Shaemus Gleason from Clarity Pharmaceuticals. And we're ready whenever you are, sir.
All right, great. Well, first off, thanks for everyone for joining. It looks like we have 121 participants today, which is a great turnout to talk about copper-67. So let me just share my screen. Presentation mode. All right. My name is Shaemus Gleason. I'm the Executive Vice President of U.S. Operations for Clarity Pharmaceuticals. We're going to do a quick update today on what we've accomplished in the last few years at Clarity Pharmaceuticals. And thanks to Alan and Brian for mentioning early on kind of some of the advantages of the CoSAR chelator. So what does Clarity Pharmaceuticals do? The whole company is predicated on this idea that if you could leverage a unique chelator that was capable of harnessing both copper-64 and copper-67, could that be applied for a unique theranostic approach in the space of nuclear medicine? And so that's what Clarity does, right? We're the leader in targeted copper theranostics. We're the only company with a clinical phase copper-67 based program. We have a proprietary sarcophagan chelator that was previously mentioned that has some advantages already discussed. We have a few assets in the clinic today that I'm going to go through, some good patent families covering this IP. And more importantly, too, we have a really experienced team. I think everyone on this call would agree that availability of isotopes is one thing, but the availability of talent to develop these isotopes is also a hindrance for new companies. And so what Clarity has is very unique is we have a collection of different skill sets with large experience in this space. You know, we have a lot of Dota alumni here who have successfully developed and commercialized compounds. So, you know, we're very happy about that. And the other thing about copper that we'll touch on at the end is has some long-term commercial advantages potentially. So we've went through this a few times, but we'll go through it again. So this is a theranostic approach. We harness copper-64, the PET agent, for the diagnosis of the disease. And then we come behind with a copper-67 based beta particle to treat said disease. So why is our chelator different? What you see here on this slide is a comparison between copper-64 sartate and copper-64 dotatate. In these images, you see the inherent issue with dotatate-based chelators, previously Dota-based chelators, in the actual clearance of these products. And so the copper-64 images up here, you don't see any liver uptake. That's because, you know, in the body, copper-64 is well contained within that chelate. This isn't true for copper-64 dotatate. You see, you know, liver uptake, which can be problematic, more so in a therapy, right? You don't want liver uptake. So that's what we see our advantage of being for our platform. This is another kind of analysis. And so what we have is high accuracy and high precision. We say high accuracy because we only treat patients who show uptake in the tumors. So this is an example of how we do our trials. We look at, do you have a positive uptake for our copper-67 based 64 base vector? And if so, you continue on to copper-67 based therapy. If you don't, you go on to conventional therapy. This is a picture from a trial we did down in Australia named meningioma, which shows exactly this, right? This is a copper-64 scan. You see uptake here. We later come behind with a copper-67 based therapy. You see uptake in the exact same position. This is what you want to see with the theranostic. So this is our pipeline today. We have SARTate, which is being investigated in neuroblastoma, and Alan kind of hit on some of the key points there. We're also investigating the SARTate SSTR2 target for neuroendocrine tumors. We have a bombesin program that I'm not going to talk a lot about today, but we have an ongoing trial down in Australia looking at a bombesin diagnostic with the intention to pursue it as a theranostic. And then more importantly, and probably the big ticket item today, is we actually are recruiting today a SAR-BIZ PSMA prostate cancer trial in the United States. And we're going to talk a little bit about what BIZ PSMA is and the potential advantages. So I want to show some pictures today. This is the CLO4, right? This is our SARTate trial. This is being done to dose escalation expansion in high-risk neuroblastoma patients. The trial is currently recruiting. We're in the dose escalation phase. Memorial Sloan Kettering Cancer Center is a member of this trial, and we have a few additional US sites. And so talk a little bit about accuracy. Alan already covered the neuroblastoma kind of disease state. So one of the things that we see as an advantage is we have high accuracy. So this is the same patient looking at an I-123 MIBG scan versus our copper-64 SARTate scan. And you can see, these images look much different. Followed up by that is we have high precision. So this is the same patient in our dose escalation trial. And we see copper-64 SARTate. This is the screening. We see uptake in these locations. Later on, we do a copper-67 SARTate spec image, and we see uptake in those same locations. So that's a very good thing to see in a trial, and we're encouraged by that, and we continue to recruit actively into this trial at Memorial Sloan and other institutions. This is probably the most exciting slide in the presentation, and the reason for that is we've just gotten underway on this trial in the United States. So what Clarity did is they approached the PSMA problem from a different perspective, so if you look at, for instance, lutetium PSMA 617, they have one targeting moiety. What we were able to accomplish is add an additional targeting moiety, which sounds simple, and I'll show you the results in a second, what that means. So these are some quick kind of preclinical biodistribution studies. What you see is what you'd want to see, right? High uptake in the tumor and consistent uptake, clearance from the spleen and clearance from the kidneys. On the bottom here, you see we have a dose-dependent response curve, which is also what you'd want to see. We looked at 7.5 megs of 67, 15 megs, 30 megs, and then a fractionated 15 by 15 meg schedule. Another ideal characteristic of a theranostic is you want to see clearance from the kidneys. This is what you see in this mouse model. The kidneys are pretty hot at 1 hour, but by 24 hours, most of the copper-64 is cleared away. And probably the most exciting slide that we have, and one of the things that I'm most interested in, is this is looking at the percentages of adjective activity for PSMA 617 versus our SAR-BIZ PSMA. And what you see is almost twice the tumor uptake of the BIZ PSMA as opposed to the kind of phase 3 investigated PSMA 617. How this plays out in the clinic is to be determined, but we're very happy to be in this graph as opposed to that graph. So this is a little about the ongoing trials we have in the prostate space. Our theranostic trial is called the SECURE trial. It's a phase 1/2A, looking at using copper-64 BIZ PSMA and copper-67 BIZ PSMA for identification and treatment of PSMA expressing prostate cancer. So we're doing a theranostic multi-center single-arm dose escalation study with a cohort expansion for up to 44 patients. We have an open IND currently with the US FDA for both these agents. And what this does is empowers diagnostic PET imaging with 64 selection patients suitable for therapy for 67. This trial is currently recruiting and can be found on the clinicaltrials.gov website. The other trial we're running right now is called the Propeller trial. This is being done down in Australia, and it's a phase 1 blinded review dose ranging study of 30 patients in Australia. What the disease state this is in is it's once you're diagnosed with prostate cancer and looking at pre-radical prostatectomy patients. And what it's doing is determining the safety and efficacy of the 64 BIZ PSMA, and then comparing that agent to gallium-68 PSMA-11, which is the current standard of care in Australia. A trial is also currently recruiting. I want to spend a few minutes because this has been hit on a few times during the presentation. It's really important for everyone to understand on the call today, if they don't already, that copper-64 and copper-67 have some really unique production characteristics that could, in the long term, lend it to some pretty easy commercial scalability. The first is copper-64 is produced on a cyclotron, and we don't need a series of regional cyclotrons like F18. We can produce the copper-64 market with some centrally located cyclotrons. And the reason for that is the 12.7-hour half-life facilitates central manufacture. And the reason we can say that with a lot of confidence is there's another product on the market, and it's been on the market since probably before the Department of Energy was the Department of Energy, and that's I-123. So I-123 has a 13-hour half-life and is provided centrally across the US and has been for a very, very long time. So that's why we have confidence that we can supply this agent centrally. And we also have a very long shelf life. Gallium-based agents have a 4-hour shelf life. We're looking at least a 48-hour shelf life for our product. So that allows us flexibility and scalability and an easy ability to bring these imaging agents to market. On the 67 front, we use electron accelerators similar to the DOE's to produce our copper-67 for these trials. Clarity currently has supply agreements in place to provide sufficient scale and suitable price points for later commercial and late-stage trials. And this is increasing, every day with the advent of rotatrons. The half-life of 67, which has been mentioned a few times, being 2.67 days, is actually really favorable for central distribution. You can get anywhere in the US with a finished drug product ready to administer. We actually do that today, even in our phase 1 clinical trials. From the end user perspective, some of the advantages of 67 is there's no long-lived contaminant or waste products like you see with the carrier add lutetium. We don't have issues with reactor outages, and we have domestic US supply at sufficient scale. So going back to kind of the manufacturing advantages, which is really important, right? Because these drugs can be developed, but if you have access to them, they don't really make a lot of difference for patients. We have readily available supply. The target tree we use is also pretty accessible as opposed to like ytterbium-176. And also kind of interesting part about our platform is because this is a true theranostic pair, and because we're using 2 copper isotopes, the actual production chemistry is very similar. So tech transfer, validations are a lot easier to pull together because a lot of the methods are shared. So that allows us to scale production at pretty high speed at low cost. And that's the end of my presentation. I'm looking forward to the upcoming panel discussion.
All right, thank you. Very exciting progress that Clarity has been making.
Thank you.
All right, I'm going to ask all the panelists to show themselves. And then now we have an open Q&A session. Brian's actually already been going through and answering some of these questions. We're going to revisit some of those. So I'm going to jump in right away and get some of these questions that are inside the Q&A function. And this one goes to Dr. Shively. What were the estimated tumor dose in the mice that you treated?
Yeah, so at 500 microcuries, we could estimate that we were getting somewhere in the range of 2 to 5 gray.
Okay. I'm going to follow up with a question of my own for you, Jack, while we're there. So with your, did you happen to image your mice that you injected with the copper-67?
No, it's kind of a sad story. We had an Invion that we replaced with a GeneX PET imaging system. And so with the GeneX, we can do CT, but we can't do SPECT. So we'd have to rely on the copper-64, which is fine.
Okay. Okay, thank you. That's disappointing, but we understand those challenges.
Yeah, we got a new instrument, but we took a step back in terms of SPECT. Yeah.
I believe that this question is for Brian. says, great presentation. We have tried pre-conjugation with 4 TCO per antibody using TZ Copper-64. Unfortunately, we have observed 10 to 20% conversion only. We use PBS buffer with pH 7.4 for the reaction. Is this conversion fine for pre-targeting imaging?
Yeah, so I seem to be stuck in a black void. I can't understand why, but it's probably for the best because I would just make everybody jealous because I'm at the beach. So, so to answer your question, no, that doesn't sound great. But it also sounds like the type of question that probably isn't of the most interest to everybody. So to answer your question, I will type my email address into the Q&A, and then you can why don't you send me an email outside of that, and we can work through what's going on. Because that sounds like something's not working right, but we spent, I don't know, 7, 8, 9 years doing this. So it's what keeps me up at night other than my 2-year-old. So I can probably help figure things out.
Very good. Well, since we have you, Brian, I think we're gonna ask you to answer some of the other questions that.
Sure. Again, sorry for the black void.
Yeah, not a problem. So one of the questions was, what was your thoughts on internalizing MABs as a targeting ligand?
Yeah, so we thought that would be a bigger thing than it ended up being as well. So obviously, if you have something rapidly internalized and you inject it, the antibody TCO goes into the tumor. So by the time that the radioligand with the tetrazine flies by, you're out of luck. That is true. And we've shown it's reasonably true for rapidly internalizing things like trastuzumab, for example. But we've also seen, and we published this, I think in 2016 or 2017, and I think actually Dr. Lappy's lab down at UAB wrote a comment on it for Journal of Nuclear Medicine. Amongst our best pre-targeting results are with this antibody called 5B1, and it targets CA19-9, and it is internalized, not overwhelmingly quickly, but it's internalized for sure. And we see pretty decent results with that, or not pretty decent. We see great results for that. So I wouldn't say that a completely non-internalizing antibody is a prerequisite here. But so something that's internalized relatively quickly, probably not the best, but in the middle is fine to work with. Generally, these things, we found that these things are kind of things that we have to deal with on a case-by-case basis.
And then following up with that question along the same lines, do you see any competition when administering the repeat doses?
We don't. So what we found, and we published this in 2019 in Bioconjugate Chem and 2020 in PNAS and in different studies, is we've seen that the injection of when we do this theranostic pre-targeting, where we inject the antibody, then inject a PET radioligand, then wait a little while and inject a therapeutic radioligand, we see that the uptake of that first radioligand in the tumor is about the same as the uptake of the second radioligand in the tumor, which is surprising. And I can see why you'd ask the question, because you'd think that the first one would take up all the seats on the bus, and then along comes the second one and he has nobody to play with. And that's no fun for anybody. But what we've seen is when we do some back of the envelope calculations with a couple of reasonable assumptions, but assumptions nonetheless, we think when we do these 2 injections, we think the first radioligand occupies anywhere from 20 to 40% of the transcyclooctanes at the tumor site. But that still leaves 60 to 80% left over for the second injection. And so that's why we think that the first radioligand doesn't attenuate the uptake of the second radioligand. We'd love to do more optimization studies here, because it would be great to get both numbers up to 50-50, or actually try a third injection for fractionated therapy. But I mean, as you guys can imagine, these longitudinal therapy studies and biodistribution studies ain't cheap. So we kind of have to go with the cash we have on hand.
Gotcha. So Brian, through the chat, you said that you had a question for Alan. Do you want to ask that now?
Yeah. Hi, Alan. How you doing? Sorry, again, black void. I just waved. You can't see me. So the question for you was, how do the tumor dose and therapeutic indices of those peptides you were talking about, how do those compare to like the iodine, the I-131, 3F8, or 8H9 that Naikang Chung is doing over at Sloan?
Well, the short answer is that we didn't do the dose of it. We didn't calculate the radiation dose from these. We've got the data, but we haven't calculated it yet. So I was thinking that actually as that question came up for Jack in the earlier thing, but we haven't done that. We have a physicist who's working with us, but we haven't gotten around to doing that yet. But that's an interesting question because as I said, I suspect that with the antibodies, we'll get higher uptake in the tumor than we do with the peptides.
But I mean, if your therapeutic indices are better, then the absolute tumoral dose is moot, and you could inject
Exactly.
more radioactivity.
Yeah.
Cool. All right. Thank you.
All right, Ellen, I have a question for you. So with the beta emissions, they usually have a little bit longer range in tissue. So I was surprised that with the response that you saw, the smaller tumors had a better response than the larger tumors. Can you comment on that?
Well, that's Jason and I have actually talked quite a bit about that. We may be reaching the point of no return, which is sort of the flip side of the question about going to alpha emitters, because the tumors were 1 millimeter tumors, and that path length is about that same size. So we're talking about going through versus, so there's that balance. And the larger tumors at the 4-week incubation may be a bit too large or too extensive to deal with. But the question is how much, which goes back to Brian's question, how much of that dose is actually delivered to those tumors as they get smaller and smaller. As they get, there's that trade-off between beta range and tumor size. The flip side of that question is some of the other presenters were doing subcutaneous tumors, which are somewhat larger, up to 5 or 10 millimeters maybe. So then if you're doing that with Copper-67, you've got relatively small range. So you're just getting the outside edges of those guys.
Okay. All right. There's a question I think all of us should probably answer. So what are the trace metals that interfere with the chelation of Copper-64 or Copper-67? Shaemus, why don't you tell us something about the sarcophagenes first?
Yeah, no, so the sarcophageine chelators are really great, but they are sensitive to radiometal impurities. And so our specifications are pretty tight. And our experience thus far with the production processes is they're robust enough that trace metals don't really show up too often. But, some of the ones off the top of my mind are iron, manganese, zinc. Those are the three kind of main ones that come to mind. But yeah, we keep a close eye on our spec for trace metals. But that being said, we don't have, how do I say this in a public way? We don't have a lot of batch failures related to trace metals. So we're pretty confident that the existing production technologies and target purification processes mitigate those risks.
And then how about Jack and Alan? You guys want to talk a little bit about Dota?
Well, we don't use Dota, but we haven't seen any problems with with trace metals in the sarcophagene studies we've been doing, and we didn't see them in our other studies with Dota. The peptides tend to be relatively less sensitive to trace metal contamination than antibodies. Jack, do you have any thoughts on the?
Well, I mean, I guess the main thing to say is that imaging with copper Dota antibodies, at least in our hands, has been very good. You can see that there's an advantage with the peptides with the faster clearance and the liver uptake. But the liver clearance of copper Dota antibodies is actually allows you to see better the liver mets. So there are some advantages to copper Dota in terms of metabolic clearance in the hepatocytes, say, versus the tumor mets.
So the other thing that I've read in a few papers was the in vivo exchange of copper and Dota with, say, iron. So So one of the things I was curious about, again, was that SPECT imaging of your mouse to make certain that it actually showed up to where it was supposed to be. And then the liver images that Jennifer showed, I think were pretty telling. Jennifer, do you have any thoughts on the exchange of copper in vivo with a Dota complex?
Interestingly, no, I was writing that down to go look at that after this meeting. So,
Sure.
sorry.
All right, well, we can get back to some of the other QA questions. All right, this one's for Brian again. Brian, it looks like the pre-labeled Copper-67 antibody had similar anti-tumor effects as the pre-targeted version. Were you able to determine the radiation exposure dose differences between the two approaches?
Yeah, it's like I planted somebody in the audience. So I was just thinking about this yesterday in a different context. And now if I can pull up the stuff. So essentially, we saw that here it is, haha. We saw that the 55 megabecquerels of the pre-targeting had about the same tumor dose, the same effectiveness as 18.5 megabecquerels of the radioimmunotherapy, which makes sense because the radioimmunotherapy is going to get more to the tumor. But under those scenarios with the dosimetry, we see that the dose to the blood, for example, would be about 327 centigrade for the pre-targeting and about 1300 centigrade for the radioimmunotherapy. Or the dose to the lungs would be about 300 centigrade for the pre-targeting and 900 centigrade for the radioimmunotherapy. So yeah, Kevin, you're totally right that the one that they look about the same efficacy, but the key for us is by keeping those radiation doses to healthy tissues lower, even if we have equal efficacy, even if we require a little bit higher dose, the actual doses to the healthy organs are going to be lower with the pre-targeting. So I hope that answers your question. David, you're muted. There you go.
Yeah, but I just caught that. New fandangled technologies. So Brian, since we still have you, we're going to ask you a question. Would theranostic pre-targeting require an independent staging scan? How would you make a treatment decision based off of Copper-64 scan in an orthotopic tumor? Total activity per lesion, would this miss smaller mets?
I guess. I mean, I think you'd need to do a whole lot more optimization and in vivo validation here, and eventually you'd need to be able to do entire clinical trials in which you see, in which you actually in patients see if the staging scan of pre-targeting gave an SUV of X, was there a therapeutic effect? And so you'd need a whole clinical trial to kind of suss these things out, just like you need a clinical trial now to see if the patient has this uptake of gallium net spot, they're going to respond or not going to respond to Lutathera. And so on the clinical level, you'd obviously need to do clinical experiments there. As far as animals go, I mean, I think the basic way that we laid it out so far is kind of how you do it, is once you know enough, once you've done enough optimization and validation, you do that first staging scan, and that is where you make your decision. Is based on your previous experience, the uptake at that point high enough such that you'll get therapeutic effect with the therapeutic radioligand or not? And in our N equals 10 cohort of that therapy study, we saw that the threshold was about 6 kilobecquerels in the tumor. But that was just an N equals 10 longitudinal therapy study. If we did this, we would want to do orthotopic tumors or syngeneic models and hundreds of more mice before I could tell you with any level of certainty, even in a mouse, that the cutoff is X. And then humans are a different story entirely.
Right, Brian, and I think that's a really good point, right? Because I think where this field is going is trying to figure out the most appropriate control variable for response. And that's something that at Clarity, we spend a lot of time thinking about. Is it, Is it how the image looks? Is it some of the underlying tumor biology? Is it the associated dosimetry based on lesion size? And it's really great that I think this field is going in that direction, because the end result of that could be saving patients from having to undergo therapies that won't be effective for their particular disease, and that has advantage both to the patients and to the larger healthcare system, right? There's no sense in administering patient therapies if it's not going to have any effect. So yeah, we really appreciate the work that everyone's doing here, trying to suss out, what the most important control variables are for response.
I totally agree.
Well, since you're talking, Shaemus, I have a question for you.
Cool.
Now with the BIS PSMA, I mean, you're making the molecule bigger and bigger, and now this changes the lipophilicity and everything else. And my question is, how long does it take for that BIS PSMA to distribute,
Yeah.
and localize?
So I'm going to have a much better answer to that question because we just started our human trials. We're currently underway, but our expectation is still a small molecule, right? It's just a slightly larger small molecule. We don't expect a lot of difference, definitely not an antibody. But I'll be able to give a really appropriate answer to that question in maybe the next few months. But for right now, I would kind of hold off and wait till we have some good human clinical data to share.
Sure. And then a follow-up question to that is, since the PSMA is working out so well as a BIS, would you consider doing other, right? Like the Dota tape, well, not the Dota tape, but the SAR tape with a BIS tape?
So that's a great question, right? And I think that's one of the strengths of what the folks at Clarity are doing, because we have a skill set that spans 80 some odd years in radiopharmaceuticals. Is now that we have accessibility for a bifunctional chelate, a BIS chelate, where we can use different targeting moieties or different vectors on either side of the chelate, we're investigating some pretty novel and interesting ideas around how to tune these vectors, leveraging our kind of unique ability to have two things on one therapeutic radioisotope.
And then do you notice any difference in the labeling yield or the conditions with the larger arms on the side?
Yeah, I can't share specifics on the call here, but let me just say the production process for these sarcophagan chelates for BIS PSMA is about the easiest thing I've ever seen in radiopharmaceuticals. It's fast, easy, and at room temperature. And the differences between a BIS and a single targeting moiety is not meaningful.
Very good.
Just to build off that, it's a little bit of a Midwesternism for me, given that's my roots. But in my lab, we always refer to it as falling off a log. So whenever somebody new joins the lab, the first thing that they radio label is that tetrazine sarcophagene, because if that can't label with copper-64, copper-67, then we've got bigger problems with the graduate student or postdoc. So that is like our baseline for what the easiest possible radio labeling could be.
Very good. Well, hopefully that log's not too big then. Jack, we have a question for you. What do you think the reason could be for the poor response that you saw in your studies? Did you do that study in immunocompetent mice? And are you planning to improve the study in any way to expect better response?
Yeah, that's, it was very disappointing because we, based on previous Y90 studies for RIT, we thought we would get a nice result with the Copper-67. So there's two possibilities that affected the study. One is that the specific activity of the neurotensin derivative was not high enough, and that's a general problem with peptides. You have to get them really hot because not only do they clear rapidly, but there is an issue of biouptake, as kind of what I was showing with the difference between NSG and nude mice. So yeah, in both cases, all of our studies were in non-competent, immune-competent animals. So that would also be an interesting thing to try. One of the things that has kept us from doing more studies on this, trying to improve, is the success with the actinium, because we don't have a generous donor right now to pay for all of our animal studies and radioisotopes. So we have to be a little bit more choosy, and of course the graduate student wants the studies that are going to succeed. That doesn't mean that I don't think that the Copper-67 has a role in the peptide field, as you've already seen. So that's why I think it was kind of fun to see the dotatate studies and so on. So I think there really is a role, and I think that the higher specific activity that's available now for Copper-67 to start with is really important. So yeah, if we do decide to repeat the study, we will definitely try to get labeled product that has a higher specific activity and see if that works. The other issue was these were slow growing tumors, which normally they grow faster. So there might have been some biological variation from a year earlier when we had faster growing tumors. But in general, we've seen that transfection of the neurotensin into the prostate cell lines does slow the tumor growth. So the general feeling is that neurotensin receptor expression is bad. But at least in these animal studies, you do get some effect versus, let's say, the tumors that don't have neurotensin receptor. So it was a disappointment. Whether or not we continue along the road for neurotensin depends on clinical studies, because the next thing we want to do is imaging in neuroendocrine tumors, because once they recur after dotatate types of therapies, they often are high for neurotensin receptors. So that's the way we're thinking right now. So if we ever do move into Copper-67, say, as a replacement for lutetium-177, this would be important to get the animal model working again. But first, we have to get through the imaging studies.
I'm glad you brought up specific activity. I'd like to go back to my question at the end of my presentation. And there's also a question here from Uli Custer asking what is an acceptable ratio between Copper-64 and Copper-67? So if copper is produced by the P2P path from Zinc-68, you co-produce Copper-64. So is this a viable route? Performing the P2P? Is this something that people are interested in because it's available? And what is the allowable ratio? Feel free to jump in, anybody. Or because you can get it as free Copper-64 from the nickel route, and you can get now the photonuclear without the co-production, it doesn't matter. I think that's what people's going for. All right, we'll skip that one. All right, so let me go back to my original question. Now, 200 curie per milligram, end of bombardment Copper-64 is apparently equivalent to approximately 40 curie per milligram Copper-67. So what is an ideal, or what is the minimum specific activity that you need in order to radiolabel these molecules and make sure that they perform the way that they're supposed to perform?
I'm going to personally take a pass on that question because our spec is a trade secret. And so if I got into that, I might get myself in a little hot water. So unfortunately, I'm going to have to take a pass on that. But in the end, the message I think that was shown by Ho in his 2021 paper is, higher specific activity with Copper-67 seems to work out better. And so, my opinion is the higher the better. But around the specifics, unfortunately, I can't really get into that.
Sure, appreciate that. Alan, I saw you smiling.
Well, I was trying to think of the answer because we've run into some issues with Copper-64, lower specific activity Copper-64, and I was smiling because I can't quite remember what the number is, and I'm going to put Jason on the spot and see if he can remember what we ran into, because we've not had any problems recently with the WashU material, but and then translating the numbers from curies per milligram to megabecquerels per micromole is tricky too. So And I can't answer the question and do that math in my head at the same time. So Jason, can you enlighten us on what the problems we were having with the 64 before? Because that gives us a sort of an edge to that.
We had difficulties when the specific activity was lower than 100, and that was the reported number. Bearing in mind, of course, we get it, you know, the next day, we get it on the Wednesday morning. We had difficulties with radiolabeling antibodies, which is what you were referring to earlier, but we haven't had any issues with labeling peptides because, of course, of the difference in mass. So as a rule of thumb, lower than 100 could cause difficulties.
Is that specific to the 64 or both?
With the 67, we've only used that with peptides. So that was a difficulty we only ran into with the 64.
Right, and it seems to, like with the chart that I showed, is the half-life obviously makes the biggest difference in what the specific activity really needs, right? The longer the half-life, the lower the theoretical specific activity is. Like for copper-67, the theoretical is around 755 curie per milligram, while copper-64 is upwards of like 13,000. So those are interesting things that I find as a production scientist interesting, and I wanted to pose that question here. Any other comments on that, or should we move on to some other questions? Higher is better. I appreciate that, Shaemus. I agree with as pure as possible, right?
Yeah.
But I have heard that sometimes you need to have a metabolized, an extra metabolizing factor, right? So you inject twice as much as what you really want to make it to the tumor, as half of it's going to get metabolized. How true is that in your experiences?
I'd imagine that's going to change from tracer to tracer to tracer. I mean, some things are bricks, generally speaking, and aren't going to get metabolized, and some things are. I mean, I don't think. I mean, all of the people here have more experience in this than I do, probably, but I would bet that a hard and fast rule about half of this or half of that is probably a oversimplification, because there are probably going to be tracers in which 9 out of 10 molecules are metabolized, and you need 1 out of 10. But I don't know, maybe Alan and Jack can bring their experience to that question.
Well, we haven't really run into it. Go ahead, Jack. I'm sorry.
Oh, that's okay. I was just going to say that, you know, we always cheat by giving our tumor uptakes as percent injected dose per gram. And so you need to look carefully at how many milligrams the tumor actually was. So when you start looking at absolute uptake, even under good circumstances, you might only see, let's say, 3 to 7% of the absolute dose going to a tumor. So it's a relatively inefficient process to begin with. And so I think that that's probably what you really want to look at. And then, of course, you already mentioned, Alan, the tissue path length of the emission. So the bigger the tumor is, actually the worse the therapy is going to be with most of the beta emitters. Although in mice, you have a big advantage over humans in terms of that particular part of the story. So yeah, I mean, I think there's a lot of factors that are involved here, and comparing studies across the board where you don't know what the tumor size is at the time of the measurement makes comparisons difficult.
I'm going to bite on that, Jack, and ask you, do you think that in response to your initial point that we kind of quote unquote cheat by using percent ID per gram, do you think percent ID would be a better thing to report?
Yeah, I think we should always report both.
So we typically put ID per gram in the paper because that's what everybody wants, and then put percent ID in in supplemental.
There you go.
But just if you could only report one, if you were the czar of J&M all of a sudden, and we could only get one, and you waved your magic wand over the entire fiefdom of nuclear medicine, what would you choose?
Well, I would say if we're going to report absolute uptakes, then
Okay.
there should be a column for the size of the tumor, plus or minus the standard deviation of the measurement too.
Okay. That's a good
Then
point.
let people calculate it themselves, but don't hide the absolute uptake because that's like when you start working with tumor to blood ratios. Now you have another denominator problem because we can get very low blood levels with some fragments and small molecules.
Mhm.
And so it greatly inflates the tumor uptake. And so people who only report tumor to blood ratios in their papers, if I were the czar, I wouldn't allow that.
I think we should start having group discussions on what we'd all do if we were the czar. Okay, great. Thank you. Sorry for barging in there.
Oh, that was perfect. That's what this is meant for. It's to have these types of conversations. But since you're talking, Brian, we got another question for you.
Since you like to hear your own voice, Brian, talk more. All right, great.
You're not a professor, are you? Yeah, of course. All right, what do you think the CMC challenge from FDA on pre-targeting radiopharmaceutical design?
Yeah, so it makes it, there's no doubt here, it makes it more complicated. Is that so for like the pharm tox study for this upcoming first in human trial, we, instead of doing the pharm tox on just a single agent, we had to do the tetrazine alone, the antibody transcyclooctane alone, and then the clicked combination. So it increased the cost of the pharm tox study from threefold. And so, but I'd argue that just like when they started doing the first DFO labeled antibody they put through the clinic, they had to do a complete pharm tox. And since then, now they say, hey, we took known antibodies, stuck DFO on it, it's going to be okay. And the FDA generally says, oh, that's fine. So I'd imagine once we do more antibodies labeled with TCO, then that'll become more recognized as a benign modification to an antibody. But ultimately, for the CMC and the FDA approval, it does make it more complicated. But I'd argue that, like, if it works and it works well, people will ignore the complication. I mean, if it is mediocre, then the complication will be a big problem. But we're banking on it working and working well, so people will look past the undeniable added complication of a two-part system.
And speaking of the two-part system, do you inject your the click or the radio-labeled aspect intravenously, or do you label it at the site of the tumors?
Oh no! If we label it at the site of the tumors, we'd be cheating. Although we'd get amazing % ID per gram, and to Jack's point, % ID if we did that. I'd love that. No, and any paper that gets me as a reviewer that does intratumoral injections has something else coming. But no, we do IV injections of both, just because if we have a patient with Mets, intratumoral injections are a non-starter.
So how much of your radio-labeled compound actually makes it to it, and how much is immediately excreted on average?
It's really hard to tell in absolute molar terms because we have to make a couple of assumptions, but the clicking yield at the tumor is about 25 to 40%.
And then where does it get excreted right away, or does it start?
Yeah, just right through the kidneys and out the bladder, and it's gone in a matter of 20 minutes.
Okay, very good. That's really nice, actually.
Yeah, it works all right.
Yeah. Yours going well.
I wouldn't be here if it didn't work all right.
All right. Well, we have a few more questions in the chat. This is a comment. Dosimetry can be used to determine a fetal dose, but also importantly determine to exceed dose to target organs. Does anyone want to comment back on this?
No, I think that's right. I mean, Jorge knows more about dosimetry than me. Although the first time I read it as feudal dose, and I was wondering what all the serfs would do with their radioactivity dose.
We need to have you on more of these, Brian. You're entertaining.
I'm enjoying myself, except for my disembodied black box over here.
You don't happen to have like just a blindfold over the top of it, do you?
I do. I actually rubbed it with my thumb a little while ago to make sure that one of my children didn't put Play-Doh over it or something. No, I don't know what's going on.
Let's see. Looks like there might be another question in here somewhere. Okay, so maybe we've already gotten to this. I think that really comes down to all the questions here. Do any of the panelists have questions for one another?
Maybe I'll just shoot right in the interest of time here. One of the things we're kind of becoming interested in here is, can we shorten the dosage regimen based on the different half-life, right? So lutetium is spaced every 6 weeks roundabouts. Because our half-life is about half that of lutetium, would there be a possibility to be able to dose at intervals that are shorter, right? Which the long-term advantage there would be shortening a course of treatment for a patient potentially by half. I'm really interested in what the panel thinks about, that. Is there any advantages to the shorter half-life of copper-67 from a therapeutic window perspective that could lead to shorter lengths of treatment time, which could have obviously advantages to patients long-term?
Good question.
Big silence. Maybe Jorge needs to answer that question. He's the dosimetry guy in this collection.
Yeah.
Well, maybe type it in
maybe the next meeting we can address this.
So he said in the Q&A answers, usually the reason for the 6 to 8 weeks is to allow for marrow recovery.
Yeah, so that makes sense in terms of the what you see for marrow dose versus with copper versus lutetium. What sort of side effects you're seeing in the therapeutic doses in humans with copper-67?
Yep.
All right. Well, I think that are all the questions. I'd like to thank each one of the panelists for joining us and entertaining all these questions. And congratulations on the exciting work that you're all doing. This is really good. I think it'll advance copper-67 much further. Again, if anyone has any additional questions, please feel free to reach out to the NIDC at isotopes.gov. They can either get you in contact with the panelists, or maybe we can answer your questions another way. So once again, thank you all very much, and we look forward to all the future work that we're going to be doing.
Thank you, David.